Good morning, students. Welcome back to Pharmacology Made Simple. In today's class, we will be discussing about the next topic, that is about pharmacodynamics. What is pharmacodynamics? Pharmacodynamics is the study of the actions of the drugs on the body and their mechanisms of action. That is, to know what the drug does to the body and how do they do it. Next, we see about the steps of drug action. The drug acts in our body through various steps. One is stimulation, depression, irritation, replacement, anti-infective or cytotoxic action, and modification of the immune status. Now let us see what is this one by one. Stimulation. It is the increase in the activity of the specialized cells. Example, adrenaline stimulates the heart. Okay, so when a specific drug stimulates the specialized cells, that process is known as stimulation. Next action is depression. It is the decrease in the activity of the specialized cells. Example, quinidine depresses the heart, barbiturates depress the central nervous system, and some drugs may stimulate one system and depress another. Example, morphine depresses the central nervous system, but it stimulates the vagus system. Okay, so here what is happening? The drug, it will decrease the activity of the specialized cells, thereby suppressing the action, the normal action of those cells. Next is irritation. This can occur on all types of tissues in the body and may result in inflammation, corrosion, and necrosis of cells. Okay, so sometimes the drugs can cause the irritation in the body, especially in the gastric mucosa, and that is why many a times we are prescribed the drugs to decrease the gastric acid so that the irritation and inflammation will decrease. Next, replacement. Drugs may be used for replacement when there is deficiency of natural substances like hormones, metabolites, or nutrients. The example is insulin in diabetes mellitus, iron in anemia, and vitamin C in scurvy. So, anytime when the natural hormones or enzymes and nutrients are decreased in our body, so drugs can be given to replace these substances. The next process is anti-infective and cytotoxic action. Drugs may act by specifically destroying infective organisms, example penicillins, or by cytotoxic effect on cancer cells, example anti-cancer drugs. And the last process is modification of the immune system. Vaccines and sera act by improving our immunity, while the immunosuppressants act by depressing the immunity. Example, glucocorticoids. Okay, so in case of cancer cells, we might require the immunosuppressants, but generally the vaccines and sera are given for improving the immunity. So these are the steps of the drug action, that is stimulation, depression, irritation, replacement, anti-infective and cytotoxic action, and modification of the immune status. Next we go on to the mechanism of drug action. The fundamental mechanism of drug action may be through receptors, through enzymes and pumps, through ion channels, by physical action, by chemical interaction, and by altering the metabolic processes. So, here we can see the image. It could be through the enzymes, it could be through the ion channels or receptor pumps, or it could be through some physical or chemical interactions. So first through receptors. Drugs may act by interacting with the special receptors in the body. So the receptors can either take in the substance or there could be the blockage of the substance as well. Next, through enzymes and pumps, drugs may act by inhibition of various enzymes, thus altering the enzyme media reactions. Example, allopurinol inhibits the enzyme xanthine oxidase and acetazolamide inhibits carbonic anhydrase. Enalapril inhibits angiotensin converting enzyme and aspirin inhibits cyclooxygenase. Neostigmine inhibits the acetylcholine esterase. So we can see that various drugs, it will inhibit the various enzymes and thus it will alter the enzyme media reactions. The membrane pumps like hydrogen, potassium and ATPase as well as sodium and potassium ATPase may be inhibited by drugs like omeprazole and digoxin respectively. Next, through ion channels, drugs may interfere with the movement of ions across specific channels. Example, calcium channel blockers, sodium channel blockers, potassium channel openers, and GABA-gated chloride channel modulators. So here what is happening? The drugs will interfere with the movement of the ions across a specific channel. Next is physical action. The action of a drug could result from its physical properties, like the absorption can be activated charcoal in case of poisoning. The absorption of the poisons can be activated by charcoal, then mass of the drug. So bulk laxatives like Salium and Ban can be given as a bulk laxative. Osmotic property, in case there is osmotic property, that will also help the drugs uh, for that particular problem. In case when the person is having the problem of inability to pass the urine properly, that time osmotic diuretics like mannitol can be given to help in the passage of the urine. Next, osmotic purgatives, the example is magnesium sulfate and radio opacity. Okay, so when we are going to take the x-rays, barium sulfate contrast media is injected into the body so that we can get the radio opacity of the x-rays. Chemical interaction. Drugs may act by chemical interaction, such as antacids may neutralize the gastric acids. The oxidizing agents like potassium permanganate is a germicidal. And chelating agents, they bind to heavy metals, making them non-toxic. So even the chemical interactions are very important for the mechanism of action of the specific drugs. Next, altering metabolic process. Drugs like antimicrobials alter the metabolic pathway in the microorganisms, resulting in the destruction of the microorganism. Example, sulfonamides interfere with bacterial folic acid synthesis. Okay, so generally, many of the antimicrobial drugs, they will be altering the RNA synthesis of the bacteria, thus it will result in the death of the microorganism. Next we move on to what is a receptor. A receptor is a macromolecular site on the cell with which an agonist binds to bring about a change. Okay, so here we can see the natural uh, things which can happen. For example, in the receptor side, if a neurotransmitter is coming to the receptor side, it gives a good pharmacological response. Whereas an agonist also will give a good pharmacological response. Whereas if the drug is an antagonist, it will not give any pharmacological response. Therefore, it will act through the mechanism of depression of the drug. Next, we see few important definitions. What is affinity? It is the ability of the drug to bind to the receptor. What is intrinsic activity or efficacy? It is the ability of the drug to elicit a response after binding to the receptor. So, affinity is ability of the drug to bind to the receptor, whereas what is its activity or efficacy is what is the response it gives after binding to the receptor. Next we see agonist. An agonist is a substance that binds to the receptor and produces a response. It has affinity and intrinsic activity. Example, adrenaline is an agonist at the adrenergic receptors. So here we can see the image. So there are some natural chemicals when they are binding to the receptor side, it will give the normal cellular activity. Whereas if an agonist is coming and binding, it will give the enhanced cellular activity. But in case of an antagonist drug, it will block at the receptor side. Thus, it will prevent or block the cellular activity. Next, we see partial agonist. Partial agonist binds to the receptor but has low intrinsic activity. Pentazocine is a partial agonist at the U-opioid receptors. Okay. So what is partial agonist? It will bind to the receptor but its intrinsic activity is low. Next we see inverse agonist. Some drugs after binding to the receptors produce actions opposite to those produced by a pure agonist. They are known as inverse agonists. Okay. So what is happening? It will produce the opposite reaction. Okay. So example, diazepam acting on benzodiazepine receptors produces sedation and anxiolysis muscle relaxation and controls the convulsions, while inverse agonists B carbolines bind to the same receptors to cause the arousal, anxiety, increased muscle tone, and convulsions. So you see over here, both of them are going to the same receptors, okay, but they're having the different action. Whereas diazepam, it will produce the sedation and it will cause the relaxation, but whereas B carbolines, it will cause the arousal and anxiety. Okay, so this is an example of the inverse agonist. Next, we see antagonist. An antagonist is a substance that binds to the receptor and prevents the action of the agonist on the receptor. It has affinity but no intrinsic activity. Example, tubocuradrine is an antagonist at the nicotinic receptors. So what an antagonist is doing, it will bind to the receptor and prevent the action of the agonist. Okay. Next we see what is a ligand. A ligand is a molecule which binds selectively to a specific receptor. Next we see spare receptors. Some experiments show that high concentration of an agonist can still produce maximal response in presence of an irreversible antagonist, and this was because of the presence of spare or reserve receptors. Thus, it is possible to stimulate the myocardium even when 90% of the cardiac beta adrenergic receptors are blocked by an irreversible beta blocker. Okay, so why this is happening? Because other than the normal receptors, there are some spare receptors which can help us to cause the action of the agonist. Okay, and high concentration of the agonist, when it gets attached to the spare receptors, it will cause the action to take place. Okay. Now, what is silent receptors? Silent receptors are receptors to which an agonist binds but does not produce a response. Presence of such silent receptors may explain the phenomenon of tolerance. Okay, so when we say that the person has become tolerant towards that drug, that means even when he's taking that medication, his body is not producing any effect, and therefore the person is tolerant to that drug. Okay, so that happens when the medication or the drug is binding to a silent receptor. So only the binding action happens, but it does not produce any response. Okay, so these two receptor types are important for you to know, that is pair receptors and silent receptors. Next, we see about receptor regulation. The number of receptors and their sensitivity can be altered in many situations. Denervation or prolonged deprivation of the agonist or constant action of the antagonist all result in an increase in the number and sensitivity of the receptors. Okay, so what is happening when there is constant deprivation of the agonist and constant action of the antagonist? What is happening? Increase in the number and sensitivity of the receptors. So this phenomenon is called upregulation. Okay, so understood what is upregulation? Increase in the number and sensitivity of the receptors. Prolonged use of beta-adrenergic antagonists like propranolol results in upregulation of beta-adrenergic receptors. Okay. On the other hand, continued stimulation of the receptors causes desensitization and a decrease in the number of receptors known as downregulation of the receptors. So first we have seen the upregulation where there is an increase in the number and the sensitivity of the receptors, but downregulation is when there is continuous stimulation of the receptors which causes the desensitization and decrease in the number of receptors. This is known as downregulation of the receptors. So here in the image you can see that in receptor downregulation, because of the desensitization, there is sustained agonist activity, endocytosis, recycling or degradation, then the synthesis of new proteins, and it gets incorporated into the membrane. Whereas in the upregulation, the receptor will become the supersensitized, and it will increase the total number of receptors, decrease the downregulation, withdrawal of antagonists, and it will elevate the number of receptors and can produce the exaggerated response. Okay, so hope you understood the difference between the downregulation and upregulation. Now, what are the consequences and implications of the receptor regulation? After prolonged administration, alpha receptor antagonists should always be tapered. For example, if propranolol, a beta adrenergic receptor blocker, is suddenly withdrawn after long-term use, it precipitates angina. Okay, so some drugs, when we are taking for a long duration of time, it should not be stopped immediately. The dose has to be reduced and then only it can be stopped. Okay. So constant use of beta adrenergic agonists in bronchial asthma results in reduced therapeutic response due to the downregulation of beta 2 receptors. Okay. So these are the clinical consequences. What will happen? Okay. So sometimes the downregulation can happen or sometimes the upregulation of the drugs can happen because of the receptors to which the drug will bind. Hope it is very clear to you students up till here. In the next class, we'll see the remaining topic of the pharmacodynamics. Thank you. So some of the important questions in this topic is, what is pharmacodynamics? Explain the mechanism of drug action, which can be asked for 10 marks. Next, steps of drug action for 5 marks. The receptor regulation for 5 marks. Agonist and antagonist for 2 marks. Spare receptors and silent receptors for 2 marks. If you like the video, please do subscribe and share. Thank you.